Good evening, all, and good morning to Dr. Parag. Dr. Parag works in Jet Propulsion Laboratory at the Caltech. So his interest is looking at the microbes on the spacecraft, which is very exciting for us. So I don't want to take much time. Already we are a few minutes late, so I will leave it to Dr. Parag to to take us into a journey on finding the microbes on the spacecraft and what is more interesting on that. Over to you, Dr. Parag.
Okay, thank you, Dr. Bala. So my name is Dr. Parag Vaishampayan. I work at Jet Propulsion Laboratory. It's NASA's one of prime center at California. So I'll talk about JPL later, but first of all, thank you so much for this opportunity. I was really looking forward to talking to you all on YouTube Live. And thanks to Dr. Bala and his team. And thank you for accommodating my time. This is early morning for me, and it's evening for you guys. So I do understand the inconvenience, as well as the screen sharing is not the optimum as I want. So with that onset, let me begin with the talk. The title of my talk is Bugs on the Spacecraft, because I wasn't aware, at least 10 years back, that somebody's actually worrying about microorganisms or bugs on the spacecraft, because I was a microbiology student back in India. I did my PhD from Pune, and I was only aware of certain aspects of microbiology. And that's my interest in talking today, so that students like you, I was in your shoes a couple of years back, so I want to give you a journey of mine so that you can correlate with yours and see that a microbiologist can actually work for NASA, which I was really fascinated to know that what's the role of microbiologist in NASA. So that's what I'm going to talk about. Since this is an astrobiology webinar, I will touch upon the astrobiology research as well as the planetary protection research I do at Jet Propulsion Laboratory. For some of you, planetary protection might be a new concept, and I will try my best to give you an overview of what planetary protection is all about and how does that apply for NASA missions. So let me give you a quick background about my journey, as I promised, how I get into NASA and how I started working in astrobiology field, which I was completely unaware when I was doing my PhD. So immediately after my PhD, I got an opportunity to work at National Center for Cell Sciences in Pune, which is on the Pune University campus. And fortunately, IUPA, which is a premium institute in astronomy, was right on the campus. And Dr. Jayant Narikar, who's one of the leading scientists in ISRO, who was a director of IUPA back then, he proposed a very interesting project. And as a fresh PhD, I got an opportunity to actually work on that project. And I'll explain about that project. So to get into that project, the concept behind that is about the origin of life. You know, there are different theories of origin of life, and the Big Bang theory and the natural evolution theory is one of that. But some people believe in alternate theories. And the reason behind that was, if you actually take into consideration the total span of the life existed on Earth, if you take the first 2.5 billion years, if life would have evolved into that period, immediately after that, there was a period called late heavy bombardment. So what happened during that period was our Earth was completely bombarded and completely burned by meteorites. So you can imagine meteorites hitting Earth every now and then for next thousands of years or even for millions of years and completely burning. So if at all there was any life, would have completely wiped out from the face of Earth. If that's the case, life has to start all over again to begin its journey. That means our window of time period for growing or existence of life is shortened by that first late heavy bombardment. Perfect. So sorry for the technical glitch. I hope the engineers have fixed the issue, and those who cannot hear or see the screen, I apologize for that.
Sorry from my side.
No worries. So I was talking about the planet protection, and the analogy to that is visiting somebody's house and not contaminating their house by your own dirty shoes. And that's exactly what we do in the space exploration as well. when we go to other planets, we do not want to contaminate the other planets by bringing anything along with us. That means on our spacecraft. And that's not to take care of the planet as such, but to make sure that our science exploration is not jeopardized. And that process is called forward contamination. And I will explain how we exactly achieve that. But along with that, if we bring anything back from other planets, we don't want to contaminate our own planet. We are already having problems with the viruses. We don't want to bring anything unknown from other planets and contaminate our own Earth, which is the only planet we know we can live as of now. So that is called back contamination. And we have only brought few samples from extraterrestrial, which is lunar samples, and we have brought some stardust samples. But we have not brought Martian samples yet, and I will talk about that in a few seconds. But that is called backward contamination. And our team, as a Planetary Protection, is responsible for both forward and backward contamination. And let's see how we achieve doing that. And the way we do that is, as you can see in the screen, the reason I put the screen is Planetary Protection plays a very important role because we are not only going to Mars, we are going to many, many other planets. As you can see, every line which is coming from Earth, that each line represents one mission NASA and other planets, other countries have ever sent. And as you can see, there are many, many missions to Moon. There are many missions to Venus, and most importantly, there are several missions to Mars. And my career, I started my career working on Mars missions, and I'm still continuing doing several Mars missions. One of the Mars mission I'm working on right now is called Mars 2020, or the name of the rover is called Perseverance. So the reason I put this slide is because just to give you a perspective of how many different missions we do. And when we go to different planets, we take different precautions or different measures. So here is an overview of Martian missions or journey to the Mars, because that's what I work at JPL. You guys probably remember some of the orbiters, but you remember those twin rovers called Spirit and Opportunity, and then Curiosity or Mars Science Laboratory. I have actually worked for 5-7 years on Curiosity, which is present on Mars at present as we speak, is doing science on Martian surface. Now let's see how we achieve doing that. So that is called NASA Standard Bioassay, or we call it as NSA. What we do is in NSA, we actually look for spores present on a given spacecraft. The logic and the philosophy behind this technique is if you can get rid of spores, you can get rid of all other vegetative cells, because spores are the most hardy and the tough cells. So if you are cleaning and baking and making your spacecraft clean, and if you don't find spores, then the likelihood of finding any other microorganisms is much less. So we actually calculate a probability of contamination when we land on other surfaces. So for every planet, there is a different planetary protection requirement. For example, if you're going to Moon, the requirement is much less stringent as compared to going to Mars, and even compared to Europa, which is an icy moon. So there are different categories ranging from category 1, which is the less stringent, to category 5, which is the highly stringent. So if you're bringing any samples back, you want to make sure that not even one single dust particle or one single microorganism is present on that given sample. So that's the range of, and we do that by actually going and swabbing and collecting samples from each and every part as we are building the spacecraft. So that's my job. So my team goes and collect the samples from every spacecraft component. So I'm very happy that we are one of the few people in the world who have actually touched every part of that spacecraft which is going to Mars, or which has landed on Mars. While doing that, we collect a lot of samples, and each sample is carefully and meticulously archived. So I have around 6,000, more than 6,000 bacterial isolates collected from Viking 1975 until 2020 today. And these samples are stored in systematic way, and we do find novel species which are present on the spacecraft, and we get a chance to name those novel species. I already mentioned about naming a microorganism after Dr. Fred Hoyle. And these are the different missions, and these are the number of organisms I found. Recently, I got funding from NASA to do a genome encyclopedia. So it's called Genome Encyclopedia of Spacecraft Associated Microorganisms. So I'm actually creating an encyclopedia of whole genomes coming from these microorganisms. It's a very interesting project because this will give us understanding of not only what kind of microbes are present, but it will also tell us what they're capable of. Will they be able to survive in extreme desiccation conditions of space? Will they be able to survive in extreme radiation conditions in space? And so on and so forth. So right now, we are working on this particular project. But talking about the novel species, we have named more than a dozen different species after famous scientists, astronauts, and places. One of the species we named after Sir Abdul Kalam, so it's called Kalami. Recently, I named another species after Kalpana Chawla. It's called Chawlai. I have named one species after Carl Sagan. And here you can see one species we named after phoenixes, which became the top 10 species in 2014. We have named it after Bacillus hornecki, was named after Gerda Horneck, a famous German astrobiologist. So that's a fun part of our job here at JPL, that we can name species or bacterial species after famous scientists and famous astronauts or even different places. So moving forward, one of that particular species or one of the particular organism we found is called Bacillus cumulus sapphire 32. And this particular spore-forming organism we have studied in detail. We have a whole genome. We expose it to gamma radiation, UV radiation, desiccation, cold, and it just survives. It survives far better than any other comparative microorganism you can imagine. Then we thought, what about if we expose this particular organism in actual space condition rather than lab simulation? So we took this microorganism. You can see these spores. They were put on metal coupons, and those metal coupons were mounted on these racks or a box, and that entire box was mounted outside space station. You can see here in the circle, it's mounted outside space station, and then it was left there for 18 months. So for 18 months, it was exposed to space radiation, cosmic radiation, galactic radiation, space desiccation, vacuum, solar flares, you can name it. So the reason it was left there for 18 months is because we calculated the total radiation it will experience as equal to a microorganism which will experience the radiation of 8.5 months of journey from Earth to Mars if it is sitting on a spacecraft. So that was really fascinating for us. Then eventually we brought those samples back. They came back to our lab and we studied them. And to our surprise, majority of the spores which are on the top, they survived. But those who are sitting underneath each other, we call it as shielding effect. And all those which got shielded, they survived. And that highlights the importance of planetary protection. You can imagine a 0.5 micron size of a bug or a microorganism can easily hide on a small scratch or abrasion or nooks and crannies of such a complicated spacecraft. And they can easily hitchhike from Earth to other planets. And that's the reason doing planetary protection more responsibly is very important. So we repeated this experiment by exposing a few other microorganisms on International Space Station many times, and we have fantastic results. And every slide I'm showing here has resulted into a publication. So we have published papers based on every experiment which I'm describing here. And I'm very happy to share that not only this particular organism, we have applied a whole array of molecular techniques, including DNA microarray, next generation sequencing, and bioinformatic analysis for understanding the spacecraft and associated surfaces. A few years back, I got another project from NASA where I was given a task of actually collecting the DNA from the clean rooms. So this was the first time we were collecting DNA from from the clean room itself, because it's very difficult to actually extract DNA from such clean environments. They are in picogram liter. So you can imagine one scoop of soil, you will have trillions of microorganisms in that one spoon of soil sample. But when you compare it to clean room, it's extremely low biomass, or we call it as ultra low biomass samples. So it was a challenge. It was a technical challenge, but I'm happy to say that for the first time, we were actually able to construct metagenomes from clean room. And not only that, a very interesting experiment we did and we published is looking at or differentiating which microbes are actually living and which microbes are dead. And we applied a very simple technique which food microbiologists have been using for a long time. For the first time, we applied it for the space science. So what we did is there is a small molecule called propylium monoazide, and this molecule can go inside the cells and covalently bind to the DNA only if the cell wall is compromised. If the cell wall is not compromised, PMA cannot go in. So we actually expose our samples to PMA, and if the cells are dead, if their cell wall is compromised, as you can see here in the slide, PMA can go in and bind, and that DNA is no longer available for any further analysis. So for the first time, we performed viability-linked metagenome in the world, and we applied that to the clean room. So this is the first example of metagenome done using live-dead technique. So we published that paper, and then we worked with JGI, Joint Genome Institute, which is funded by Department of Energy here in California, and we did a lot of high throughput bioinformatic analysis. And for the first time, we showed a complete picture, which includes not only bacteria, but archaea, viruses, fungi, and many other eukaryotes in our clean rooms. And this is a beautiful picture we created, giving a complete picture. Not only that, we were able to finish 3 viral genomes coming from the clean room as well. So that was a very fantastic paper, and it's already published. These are the few publications came out from that study, which is a metagenome study, and we were able to show that you can actually look much deeper into the clean rooms. And now we are applying the same techniques for the spacecraft surface itself. And I will talk about that in my next presentation, because that study is still going on, and we are working on that study. But a very exciting study going on at JPL right now. Now, shifting gears slightly, so that was my introduction to planetary protection, what I do, but along with the planetary protection, I also do a lot of astrobiology, and my field of interest in astrobiology is life in extreme environment. So I get a chance to travel around the world and collect samples from extreme environments, and I'm mostly interested in extremely hot environments and hydrothermal vent or hot springs. So I've been to Iceland, many other places, but just to name few, we've been to Iceland collecting samples from the hot spring, because the entire Iceland is, you can imagine, Iceland is no ice. Greenland, or Greenland is not green, it's completely covered with ice, but Iceland is completely covered with volcanoes and different hot springs. So we actually collected samples, hot spring samples, studied them. We've been to New Zealand, we've been to the Japanese hydrothermal vents, collected samples from there. We have several experiments on International Space Station, and 2017 we went to UK. There is a mine called Baldy Mine, which is 1.1 kilometer deep underground, and there is an astrophysics lab, because you don't experience the background radiation. You don't experience the background radiation in that lab. So we have set up an astrobiology lab there to study how life can survive in these extreme conditions there. One of the fascinating projects I was involved in was caves in Venezuela. These are the high mountains of the Puris, which is featured in some of the Hollywood movies, where you have caves on top of those mountains, and we were actually able to go inside those caves and collect the samples. And we have published papers based on that. We have actually seen a completely different phyla present in in those cave environment because they have not seen, they have not interacted with the outside world. So that gives us a perspective of when we go to Mars, what kind of life we can expect, because life was evolving completely differently for millions of years on its own. And if that happens, how life evolves and grow into a completely different forms of life. So it's a very fascinating work, but I always like to connect the dots with what we're doing in the lab and how we can apply that on the ground. So is my research, is the astrobiology research important for day-to-day life? And the answer is yes. One of the most important things we are doing is we are safeguarding the life of astronauts which are living up there on International Space Station by monitoring the surface, water, and air of International Space Station to make sure that astronauts are living in a safe environment. And we are doing that continuously by understanding the microbiology or microorganisms present in that environment, which is also true because 90% of our life we live in indoor environment. So there was a huge initiative called Indoor Microbiome, and we have our study or our research is helping creating a better conditions for living in indoor environment. Our clean rooms are also similar clean rooms in semiconductor industries, drug manufacturing industries, hospitals. So our research is actually helping, and I will talk about one success story later, but that research is also applied for hospital and pharmaceutical manufacturing. Last but not the least, eventually we will set up human habitations on Moon and Mars and beyond. So we are doing some simulation experiments by actually looking at how we can live in confined environment for long duration of time if we go to other planets. So this is an experiment we did in University of North Dakota, where student astronauts, they live in this habitat for 10 days, 30 days, and we actually did the indoor microbiology or indoor microbiome of these environments. I would like to show one success story of application of the astrobiology research for medical research, what we did at JPL. You probably know the Human Microbiome Project. So several years back, NIH funded a human microbiome project where they collected samples from different parts of humans, hundreds of humans, and collectively they created a microbiome which is 10 times more microorganisms on our body than our cells. So we have such a huge microbiome within and on us, but we actually miss some body parts while doing that because our notion was microbes do not survive, or microbes are not present in those environments, which includes brain and breast. So as you can see in this picture, nobody has studied actual brain samples or breast samples for looking at microbiome in those samples. But we got an opportunity to work into the breast cancer research. And as you can imagine, 1 in 8 women in our subjected to breast cancer risk, and there are more than 300,000 cases just in 2014. Out of those, 40,000 died because of breast cancer. And there is a huge funding in breast cancer research, but nobody has connected the dots between the microbiome and the breast cancer. It so happened that we have an initiative called JPL Medical Engineering Forum, where 2 breast cancer researchers came and they were looking at the microbiome of the breast. And we were able to apply the techniques we have developed for understanding the microbes on spacecraft, which is also an extremely low biomass environment like the breast. And we were able to actually collect the samples from nipple aspirate fluid. That's where the breast cancer starts. And I'm happy to announce that we published the first paper showing the presence of microbial DNA in the nipple aspirate fluids, as well as statistically significant difference between subjects which had breast cancer versus those which are controlled. So this is a correlation study, not a causation study, but we applied some of our technical know-hows for medical research. So that's a success story of how astrobiology research could be applied for medical research. So moving along, I believe that if I'm doing the research in my own lab, it's worth nothing unless I transfer that knowledge or transfer that enthusiasm to the next generation of scientists, which are students. So since 2011, we are running a program called Spaceward Bound, and under the Spaceward Bound, we go to a desert environment here in California, in Mojave. We stay there for 4 days along with the students. We help them actually collect samples and perform astrobiology research. Then we took this Spaceward Bound program international by actually going to New Zealand and doing the first international Spaceward Bound in New Zealand in 2015. And then in 2016, we actually performed Spaceward Bound here in India, in Ladakh. So a team of scientists from Sweden, US, Australia, and India, we were there in Ladakh doing astrobiology for 10 days. And then 2017, we were in UK in the Balby mine. And I'm happy to announce that few thousand students from Kalam School actually watched us live doing experiments down in that Balby mine. And we were interacting with the Kalam School in India, students in Kalam School in India, while doing the research in 1.1 kilometer deep in ground in UK, in Balby mine. So that's a great educational outreach activity where you involve students into the frontier astrobiology research by actually bringing them virtually on field. So that's some of the great stories. We are doing a lot of educational outreach for minorities and historically Black colleges and schools. So for the last 3 years, I'm doing a lot of workshops for engaging minority students in astrobiology research, and I'm continuing doing that. In fact, from next Monday, I'm starting a 5-week program for these students. So then I thought, why can't we do something similar for students in India? And I'm very happy to share with you that we developed a first certificate course in astrobiology in Modern College, and the credit goes to Dr. Rebecca Tombe. She's a professor. Now she's based in UK, but she was a professor at Modern College in Pune, and we conducted 2 batches, or more than 2 batches in Modern College, and trained students in astrobiology. And then in 2018, December 2018, we conducted a first international astrobiology conference in Pune, and that's where I met Dr. Bala as well, and many other scientists around India and outside. And we had a really interesting conference in astrobiology. So that's some of our contribution in astrobiology for students in India as well. I would like to thank all my collaborators around the world and in the US for their contribution, the students coming from these universities in my lab and doing the fantastic research. Last but not least, the most important is the next generation of scientists and reaching out for stars. And that's the spirit of NASA, that's the spirit of space science and astrobiology. So I'll stop here and I'll take any questions.
Thank you, Dr. Parag. Thank you very much. And we are sorry for the technical glitches in between. There are a lot of questions that are coming in, both in the YouTube live as well as to our emails. So I'll start with the first question. This first question is from Dr. Manan Desai. Is there any chance of viral contamination or any studies done on potential effect of space environment on viruses?
That's a very, very dangerous question right now. I'm just kidding. Especially now in the corona situation. But no, I mean, even though viruses were never a target, but we are studying viruses. And of course, as I mentioned in our metagenome, because metagenome is unbiased, it's not only targeting or looking at bacteria, or is looking at everything. So we have seen signatures of viruses in some of these samples. Now the question is whether they are living or dead. In any case, viruses cannot live outside. So viruses are ubiquitous, no matter what. They are present on my computer, on my screen, on my table. So they are present everywhere, no matter what. The question is what kind of viruses are present, and are they pose any threat for our space exploration? Because the whole idea of planetary protection is to make sure that we should understand what we are carrying with us and what contamination risk it poses when we land on other planets. So yes, we are looking for viruses in clean room and other associated surfaces.
Okay, thank you. Then we have another question from from Ms. Shabani Patnaik. Will be a better alternative to research on samples brought back by sample return missions on advanced lab setups in space stations, rather working on Earth?
Sure. So if I understand the question, they're asking about, is it better to work on samples brought back from Mars rather than working on samples on ISS? Is that correct?
No, the question is that instead of bringing the samples back to the surface of Earth, maybe we can work it within the International Space Station on we have an advanced lab.
Yeah, sorry, I understand the question now. So yes, we call it as break the chain. So the whole idea is that you don't want to bring something back, and we thought about doing that. But as you can imagine, space station is always habited by astronauts there, and we don't want to contaminate, or we don't want to pose any risk to the astronauts as well. And then there are limitations of what kind of science you can do, because there is a microgravity there on space station. You won't be having that kind of confinement spaces. You will require a BSL 4 level confinement for storing those samples. So just to give you a perspective, we have not brought samples back from Mars yet. But the next mission, which we are launching on July 13th, it's called Mars 2020 or Perseverance, and I'm working on that project. We will be collecting and storing those samples in small tubes on Martian surface, and the next future mission will actually bring those samples back. And then there is a very interesting concept we have developed. So many scientists who are working on Mars sample retrieval are actually working on different ideas and the way we will store those samples. So right now the plan is the next mission will actually bring those samples back on Earth. But the way we will bring back on Earth is not in one single journey. So there will be phases, and we'll break the chain so that we are not bringing anything which has seen the outside Martian surface. We will only bring the tubes which contain those samples back on Earth. They will be confined and stored in BSL 4 or even higher stringency level conditions. But eventually, you need to open those samples and work and do science on those samples. So all that architecture, all that infrastructure is in development as we speak. And a team, including some of my colleagues, are working on those. So yes, there is the idea of bringing the samples to ISS rather than bringing them back to Earth is a fascinating idea, but there are some technical limitations doing that. So we will bring the samples back on Earth and store them appropriately and safely. That's a good question.
Thank you. That's very interesting. There's one other question from Anurup Mohanty. Do you find any microbe in common between clean room samples and human microbiota?
Sorry, clean room and?
Clean room samples and human microbiota.
Oh, yes, of course. As you can imagine, we work in clean rooms. So clean rooms are HEPA filtered. That means more than 99.999% of the air is filtered. You can see in one of those screens, you can see we are wearing the complete bunny suit. So the majority of the contamination which you see in the clean room is actually from humans. So humans are the key source of contamination in the clean rooms. I mean, we cannot avoid not letting people go in because they are the ones who are actually building the spacecraft, right? So we take every precaution of covering their face and eyes and everything, but still humans are the key source. So we do find a whole lot of human microbiome in the clean rooms. And of course, we do find some soil or dust associated microorganisms. So yeah, to answer your question, a huge portion of microbes which are seen in the clean room are actually human associated microorganisms, given the fact that humans are going inside the clean rooms and they are the ones who are somehow bringing the microorganisms.
Okay, thank you. There's one question which is related to the current pandemic from Neha. Can we contribute in astrobiology research in this pandemic? Any leads on this?
Sorry, you're just
there's a question from Neha. Can we contribute in astrobiology research in this pandemic? Any leads on this?
Yes, I think the whole world is contributing with their own capacity for this particular pandemic, because it's not only for one nation, it's for the entire humanity. It poses a great risk for the entire humanity. And astrobiology is not one faculty or one stream of science, which astrobiology includes people coming from physics, astronomy, microbiology, geology. So people are coming from different fields. Everybody is doing and contributing for the research. So I know that there are groups in JPL and in Caltech and many other labs who are working on mitigation and research for detection as well as countermeasures for this particular pandemic virus. So yes, the answer is yes. People are, including my group, we are contributing in every aspect we can. And I think it's everybody's duty to work on that research. And yes, we are doing that. Because astrobiology is not one faculty. It includes everybody, including microbiologists.
Okay. So, yeah, okay. There's one question from Navanil Karmakar. How do microorganisms survive in extreme temperature conditions of hydrothermal vents and geysers of Iceland? How does biomolecule protein survive? Don't they get denatured?
Yes. So we call it as a limit of life, right? I mean, the whole idea of doing research on life in extreme environment is to understand what are the limits of life. And our notion of limits of life are based on our own limits. But for microorganisms, you can imagine it's a completely habitable environment, even down in the hydrothermal vents or in extreme cold conditions, or the river in Atacama in Spain, or extremely hot and dry conditions in the deserts of Chile. Because we believe that these are extreme environments and limit of life or protein degradation, microorganisms are actually very tenacious and very hardy. They are able to survive in these extreme conditions. I'll give you an example of barophilic organisms, microbes which survive at the bottom of the sea, extreme high pressure. At that pressure, any living being cannot survive. But when we went down to the hydrothermal vents to collect the samples, we were surprised to see that not only microorganisms, fishes, crabs, and many other higher organisms are just surviving and flourishing at that bottom condition. So life do survive, microbes do survive. They have a different technique for coping up with those extreme conditions. Like, for example, if it's an extreme acidic condition, we may believe that the protein structures will not survive in that extreme acidic condition. But microbes have developed a different cell wall or different structure or different complex proteins which allow survival and avoid the folding of the proteins in those conditions. So a great deal of microbial world as well as higher organisms have been explored in these extreme environments. And when you go to Mars, the most likely kind of life forms might have existed on Mars or could be even present now will be microorganisms. So the chances of finding unicellular microorganisms on other planets as well as on our own Earth is much higher. I think we have seen microbes in every inch on Earth. I think except magma, we have seen microbes everywhere on Earth. And they have found or devised a mechanism of surviving and keeping their proteins and keeping their cell wall structure intact in those conditions.
Okay, just a curious question. When you mentioned about magma, so up to what temperature have they seen microbes?
Yes. Right. So we have actually found out at what temperature microbes can survive. I think the highest temperature microorganisms can survive is anywhere between 110 to 120 degrees Celsius. People have reported them surviving in much higher temperature. So when we collected the samples at hydrothermal vent, at the nose or the black smokers, the temperature is above 300 C, and we don't find anything right there. But immediately next to that, once it gets diluted and the temperature drops down, it can survive. The famous example of the enzyme we get is from the same place where they were able to survive at high temperature. So microbes or spores, they do survive at these high temperatures.
Okay, thank you. That was a bit curious for us. There is a question from Dr. Sharad. Because I'm not a biologist or a microbiologist, so I could not understand that. So I've just pasted on your chat screen. Because if I read it, can you see that? If not, then I can read it. But I don't understand the question.
Is it in the chat? Yeah, okay.
Yes, it's in the chat. You can see that?
Yeah, I can see that. I'll read the question and see if I can understand. Is the conjugation, transformation, transduction process are similar as it happened on Earth, or there is any chance that the process and the expression of resilient? So he's talking about the processes in the cells here on Earth. There's a chance that in process and expression, I didn't quite understand the results, but if he's trying to ask if the same processes, so transformation, transduction, and conjugation, these are the processes by which microbes can share the genomic DNA with each other. So in conjugation, it can transfer the plasmid. In transformation or transduction, it can share a piece of DNA with each other. So that happens all the time. I think he's revived the question. There is a new question. So I didn't quite get the question, but these processes are very common processes happening here on Earth. But if he's talking about these processes on other planets, not quite sure, because we have not discovered the life on other planets. So we don't know how the mechanism or what kind of DNA structure, or even if it's DNA or RNA structure. So we quite don't know about how that happens. So I'm sorry, I didn't understand the question itself.
Okay, thank you. There's another question. I've just pasted it on your chat screen. So it's interesting.
I'll read the question first.
Yes,
is there any chance that an astronaut carries a very infective germ, like the current coronavirus, to the ISS? If any such incidences does occur, what is the course of action taken on board the ISS? Yeah, it's a very interesting question, and I get this question all the time because, as I mentioned, that we are actually studying the microorganism or microbiome on International Space Station. As you can imagine, there are any given time, there are anywhere between 1 to 4 or up to 6 astronauts are living in that confined space, revolving around the Earth, and it's very challenging. We have encountered where astronauts have reported some kind of difficulties, but there are no serious implications where you have. And the only logical answer to that is, if that happens, if it's a life-threatening situation, we do have shuttle missions which can be launched and we can bring the astronauts back on Earth. Of course, it will require some planning and time for doing that. Fortunately, it has never happened that there is a life-threatening situation for the astronauts on space station. So they were they came back on their planned written mission. They do have medical supplies so that they can treat themselves. So yeah, fortunately, and we should keep our fingers crossed that such situation should not or will not happen on space station, because these are the brave and most strongest people chosen to stay in that extreme environment. So they are anyway going through a lot of stress on space station, and we don't want to risk their life by exposing them to viruses or any other microorganism. So yeah, to answer your question, we have not seen any life-threatening situation on ISS yet.
Okay, thank you. Following that, there is a question from Manarup Mohanty. Are tardigrada a concern for planetary protection?
Yes. So I know why the question came, and this question comes all the time. Tardigrades or water bears are life forms which are extremely resistant, and I'm personally I have worked on tardigrades, so I know they can survive in extreme desiccation conditions, and they can revive when they get the right conditions. So yes, tardigrades is a concern for planetary protection, definitely. Not that we are looking for tardigrades, because on a spacecraft we actually don't see tardigrades, or we're not looking for the tardigrades. The kind of conditions we expose the spacecraft will anyway deactivate or kill those tardigrades. The question came because in one of the recent mission, not US mission, non-US mission, they actually had tardigrades for the moon mission, and it accidentally landed there on the moon. And then the whole discussion started: what happens to those tardigrades? Will they flourish and grow on the moon surface or not? And it's a real planetary protection concern for taking any such organisms, including tardigrades and other spores. So yes, they do pose a threat for planetary protection. And it's a very fascinating research to do on tardigrades. These are and they are ubiquitous. Tardigrades are present everywhere in the world. You go outside on a pond and you take a water sample, you will find tardigrades there. So yeah, it's a real question they'll pose for
So they are everywhere.
They are everywhere. Anyway, they're everywhere on Earth.
There's a question from Rebecca. What group of microorganisms is most abundant in the spacecraft assembly room?
Is it Rebecca?
Yes.
Okay.
Is it archaea, bacteria, or viruses?
Yeah. So the majority is bacteria for sure. Archaea, fungi, and viruses form a very small portion. And we have published, I think I have shown in my presentation, we have published many publications where we have looked in detail at what kind of microbial profile, we call it as, what kind of microbial profiles are present. So bacteria comprise of majority, or almost 90% or even more, are bacteria. Very small amount of fungi, archaea, and viruses are seen. There is some DNA signatures of or eukaryotic signatures are also present. And as I mentioned, they are mostly coming from the dust and the dirt particles which are brought along with the humans. So yeah, and within those also is quite a representation which is coming from dust and dirt particle. Within those bacteria is majority are associated with human-associated microbes, and others are associated with soil. So yes, bacteria forms the majority group in planetary protection samples.
Okay, just one more question, or I'll read from the GChat. I'm getting questions in the GChat. Okay, so there's another question. So when you talk about microbes, okay, what is their lifetime and what is their source of energy on a spacecraft?
I don't understand what. So, what is the life of microorganisms?
What is the lifetime of a microbes? Yes, and what is the source of energy for them?
Okay, that's a very interesting question. And so when we study the microorganisms on the spacecraft, we always think about, and actually I got a 3-year funding from NASA to study exactly this question you're asking. What kind of energy and carbon source microbes get on the spacecraft? And we are actually, there is one publication came out, um, which deals with that. So they propose that the cleaning agents like alcohol and Cleanol 60 and many other things could, and I'm stressing the word could, uh, actually contribute to the energy and carbon source for these microorganisms. Majority of the microbes are in dormant condition on these extreme environments or the spacecraft environments. They are in a dormant condition. That means they are in spore forming condition because, or they might be oligotrophic microorganisms. Oligotrophs are the microbes which require extremely low nutrient source. So even a sub-picomolar amount of carbon and nitrogen source could be enough for them to grow. Their doubling time is extremely slow. For an actively growing E. coli in the lab, it's less than 30 minutes. So every 30 minutes, they're doubling. But some of these oligotrophic microbes living on the spacecraft surface, their doubling time could be in days or even it could be in months, because they're saving their energy for their metabolic activities. So microbes do survive. We have not clearly understood how exactly they use the carbon and nitrogen sources in these environments. And as I said, we got money from NASA to do exactly these things. For the next 3 years, I'll be studying that. So hopefully there will be a publication coming out from this very same scientific question you just asked.
Okay, there is one question regarding a career in astrobiology. Okay, this is again from Anurup Mohanty. What will be your suggestion to an undergraduate student in India looking forward to a career in the field of astrobiology?
Yes. So for that same reason, Rebecca and I started the astrobiology certificate course in India, so that there will be, at least there will be an outreach and there will be an education about this new field in India. To my knowledge, there is no university was offering a master's or bachelor's level course. Pune University is going to start or has started a course in astrobiology, master's level course in astrobiology. There is a center, Amity University in Mumbai. A friend of mine, Siddharth Pandey, he has started a center of excellence in astrobiology in Amity University in Mumbai. So there are very few avenues or few opportunities for Indian students to actually get into this. There are some online courses are available. So those who are interested in understanding the astrobiology, Dr. Charles Koppel has a course on Coursera. So if you know Coursera.com, if you look for his course, it's free. That course is very informative. So those who are interested in at least getting to know about the astrobiology, they can enroll for that course or take that course online. Hopefully, we will come up with books in local languages on astrobiology. So that's the project Rebecca and I am working on. So hopefully we can bring astrobiology books in local languages like Hindi, Marathi, Bengali, and Tamil. And so that will be a great resource for students in India. But of course, for doing research, you don't have to enroll into an astrobiology master's course. You can be a microbiologist, you can be a computer science student, you can be a physicist, and still continue your research, further research in astrobiology, because everybody can contribute to this particular field. So no matter what undergrad or graduate level course you are in, you can still continue doing your research in astrobiology. You just have to find the right place to do your research and right project to work on. So don't worry if you have not enrolled into astrobiology, you can still do your career in astrobiology. I'm a microbiologist and I'm doing my career in astrobiology in NASA.
Thank you. Dr. Parag, you have time for just 2 more questions, or we are running out of
Sure.
time? Yeah, okay. One question from Anurup Mohanty once again. Does Planetary Protection Group work with all the space organizations around the world or only NASA?
So, Planet Protection Group, every space agency has their own planet protection groups. So, my group works only for NASA. So, all the NASA missions, we are involved in planet protection research. There are private companies like Lockheed Martin, Boeing, SpaceX. So, they do have their own groups, which does some planet protection, but we work very closely. We work very closely with other planet protection groups, like for ESA, for JAXA, they have their own planet protection group. We work very closely with ESA and share some of the knowledge, because the space exploration is not about competition, it's all about collaboration. And we are very open to work with ISRO as well. So ISRO is planning to send humans as well as to Mars mission. ISRO will require planet protection research, and they have to have a planet protection group and people policy. Basically, having a policy is very important. So yeah, we are very open to working with others. We support NASA missions, but we are open for working with other agencies as well.
Thank you. One question with respect to the landers on Mars and microbes. The NASA landers on Mars. Okay, the question is, do you think the microbes that has gone with the spacecraft or the landers, do they survive in the Martian environment?
Yeah. So again, this is a very common question also, and there is a very straightforward answer to that. When we do planetary protection, the whole idea is we cannot build a completely sterile spacecraft, right? I mean, it's just not practically possible. So what we do is we calculate the total microbial bioburden on any given spacecraft, and then we add different constraints. For example, doesn't matter how many microbes you have, what is the probability of contamination? That means if you take into a factor of the time it requires, what kind of radiation it will expose to, what kind of space condition it will expose to, and upon landing, what is the probability that these microbes will survive on that planet? So every microorganism or a species of that particular microorganism has a different probability of survival. And we take into consideration for that. And only when the probability of contamination is zero or even below zero, then only we launch the spacecraft. So technically speaking, the probability of contamination is zero for any planet. So I think that's the right answer to the question. People always feel like, oh, you have a microbe on the spacecraft, so you're going to contaminate the Mars. But you have to also take into consideration that these microbes, we have taken into consideration the probability of survival or probability of contamination when you actually land on the Mars. And that probability is zero. So we have calculated that risk, and only when that risk is below that threshold, NASA allows to launch these spacecraft. Otherwise, it will not be allowed to launch. So Planet Protection Officer is responsible for signing on the final launch for allowing these spacecrafts to go to Mars or any other planet.
Oh, nice. Okay, thank you, Dr. Parag. I think though there are many questions, we'll stop here because time keeps going. Thank you very much on behalf of the students and researchers in India. I hope certainly there'll be somebody who will be taking up this area.
Oh, absolutely. So thank you, Dr. Bala, for giving this opportunity. I was really looking forward to talking to your students, and I'm really sorry for the technical glitches. And
no, the technical glitches on our side. So we are sorry for that. Okay, so thank you. We'll stop the live streaming